My name is Ola Hammarsten. I'm a professor and chief physician at Clinical Chemistry Sahlgrenska University Hospital in Sweden. In a separate movie, I've presented preliminary evidence that the release of troponin from unstressed cardiomyocytes is a more or less normal phenomenon and not restricted to dying cardiomyocytes. I have also presented preliminary evidence that released troponin is taken up locally by endocytosis by cardiomyocytes before the troponin molecules reach circulation, a system I call cleaning your own backyard. Assuming that local clearance and physiological troponin release exist, what are the current evidence for this and how will it affect interpretation of troponin elevations in patients? First, troponin elevations could be due to increased formation of cell wounds. The extent of macromolecular exchange over the membrane in apparently living myocytes, likely due to cell wounds, is often measured by accumulation of intracellular albumin. Albumin is a good choice as normal albumin is not subjected to endocytosis. In this case, albumin is followed by intravenous injection of Evans blue, a dye with red fluorescence that bind tightly and preferentially to albumin. Ischemia and reperfusion result in apparently living myocytes with intracellular staining of Evans blue. An alternative is immune staining with albumin-specific antibodies and measurement of staining only in the central portion of cells to minimize background from extracellular albumin. Shown here are control rats and rats exposed to 90 minutes of 16-degree downhill running. There is a large literature on mouse and rat models that show excessive macromolecular exchange over the muscular membrane in apparently living myocytes, both in stressed and unstressed states. Albumin also accumulate in normal rat cardiomyocytes. Accumulation of intracellular albumin in apparently viable cardiomyocytes increases about tenfold if the heart is subjected to pressure overload for 60 minutes. If stressed by low doses of the noradrenaline analog isoproterenol, albumin in apparently viable cardiomyocytes increases about threefold, as seen here using both conventional microscope and electron microscope to measure intracellular albumin with gold-labeled albumin antibodies. At isoproterenol doses that result in troponin elevations, most Evans blue positive cardiomyocytes look normal, apparently because they were able to repair the membrane damage that allowed albumin to enter. One can conclude that cell wounds are prevalent in cardiac muscle both in stressed and unstressed states. As cell wounds allow albumin to enter, it is most likely that intracellular proteins like troponin escape during this process, possibly generating troponin elevations without necrosis. Especially if the troponin elevation only lasts for 1 or 2 days, as often seen in marathon runners. Second, troponin elevations could be due to slow repair of cell wounds. This is well studied in mice with mutations in genes involved in cell wound repair. Mice with mutation in a dysferlin, a gene resulting in limb-girdle dystrophia 2B in humans, have defects in myocyte cell wound repair, shown here by spontaneous accumulation of Evans blue in muscle fibers. In a dedicated cell wound repair assay, isolated myocytes from the dysferlin mouse accumulate a green membrane impermeable dye after a laser wound. It is now well accepted that human dysferlinopathias, such as limb-girdle dystrophia 2B, are caused by defects in cell wound repair. Dysferlin mutated mice also have defective cell wound repair in cardiomyocytes, shown here by the same dedicated cell wound repair assay using the cell impermeable green dye as in the previous study. These mice also have stable troponin elevations that is aggravated by age and also develop cardiac fibrosis. Among the few humans with dysferlinopathias, 75% have cardiac fibrosis, possibly because of poor cell wound repair in their hearts. Although dysferlinopathias are rare, They show that slow cell wound repair might result in cardiac fibrosis and stable troponin elevations. It is still not known if cell wound repair is slowed down by limited ischemia. If this is true, slow cell wound repair might contribute to troponin elevations following all forms of cardiac ischemia. Finally, stable troponin elevations could be due to decreased local clearance. There are many possible ways to interfere with scavenger receptor-mediated clearance. The local troponin clearance is likely due to scavenger receptor-mediated uptake, a system called extracellular proteostasis. Impaired proteostasis is tightly linked to aging. Stable troponin elevations might be a sign of impaired removal of sticky unfolded proteins between cardiomyocytes. In this light, stable troponin elevations might be viewed as a heart-specific age marker and thus explaining why stable troponin levels are so strongly linked to heart disease and death. The most obvious reason is ischemia, shown to halt endocytosis. For instance, endocytotic uptake of transferrin, a typical positive control in endocytosis studies, is halted by anoxia in cultures of cardiomyocytes. We see that because the green transferrin complex fails to accumulate close to the nucleus where mature lysosomes are located, and that is specifically happening in anoxic cardiomyocytes. Another possible reason for reduced scavenger receptor clearance is mutations in scavenger receptor genes. In humans, mutations in several scavenger receptor genes, for instance the haptoglobulin receptor, correlate with unusually high levels of creatine kinase. Creatine kinase is found in the circulation because of constant leakage from normal muscle cells. Higher steady-state levels might, in addition to increased leakage, be due to decreased clearance via scavenger receptor. A similar mechanism might contribute to stable troponin elevations. Scavenger receptor-mediated clearance involves a binding event. This binding event will be decreased if there are an abundance of competitors, such as protein degradation products. Small proteins and protein fragments are removed by kidney function, as shown here. In patients with kidney disease, small proteins like cystatin C and protein fragments accumulate and are linked to stable troponin elevations and poor prognosis. The accumulation of small proteins and protein fragments could interfere with scavenger receptor clearance because of their abundance in patients with kidney disease. Scavenger receptors only remove one protein at a time. If many small protein fragments accumulate, local clearance can become overloaded and result in decreased clearance of troponin, as shown here. Another possibility that could impede local troponin clearance is myocardial edema. The starting point is that troponin is constantly released by cell wounds, and the same cell can reuptake troponin by endocytosis. Release and uptake is therefore expected to be directly proportional to the membrane surface. However, uptake is dependent on the surrounding extracellular volume. Large extracellular volume result in fewer troponin molecules per unit volume, and hence it takes longer for each troponin molecule to bind a scavenger receptor. Extracellular volume is related to the distance between cells. If edema forces the cells to separate, local clearance of troponin will drop. The relation between extracellular volume and membrane surface drops exponentially as the distance between cells increases. If one changes the distance between cells using a simplified tissue model with 27 cubic cells forming a cube of tissue, the membrane surface per unit extracellular volume changes exponentially as shown in this graph. As the edema forces cells apart, a troponin molecule faces an exponentially increasing extracellular volume, decreasing the chance that the troponin molecule bump into a scavenger receptor on the cell surface. The actual distance between cardiomyocytes in a normal heart, 5% of the cell diameter, are in the rapidly changing part of the graph. Even small changes in the distance between cells will result in drastic change in membrane surface per unit extracellular volume and affect local troponin clearance. After limited ischemia and formation of edema, detected by routine cardiac MR, decreased local clearance of troponin might contribute to the extent of troponin elevations and may not only be due to necrotic cardiac cells. In addition, troponin elevations in other conditions with edema, such as myocarditis, might also be viewed in the context of local troponin clearance and explain how myocarditis, sometimes affecting global ventricular function, can resolve with little evidence of scar tissue. Perhaps poor local clearance in the edematous tissue contributes to the troponin elevations and overestimate the extent of necrotic cardiomyocytes in these cases. The mechanisms proposed here could contribute to the troponin elevations. Ischemia can, in addition to necrosis, result in increased formation of cell wounds, decreased cell wound repair, and impeded local troponin clearance. It is possible that all these processes work together, resulting in the final level of troponin following ischemic events. If true, this will perhaps nuance how we interpret cardiac levels in type 2 myocardial infarctions and other ischemic events. In conclusion, if correct, local clearance could potentially explain the strong link between stable troponin elevations, mortality, and heart disease, because stable troponin elevations might signify an old heart. Troponin elevations might not be viewed as a direct proof of cardiomyocyte necrosis, and hence widen the clinical perspective when a patient presents with troponin elevations.